Okay, we are now live, and we have SDS's own Aloyce Mwambwiga, who's going to be giving us a presentation on plant residues on stone tools from Oldupai Gorge. Aloyce is a PhD candidate, and he's originally from Tanzania. I think it's going to be a fantastic talk, so take it away, Aloyce.
Thank you. Hi, everyone. As Fege has introduced me, my name is Aloyce Mwambwiga. I'm a PhD candidate at the University of Calgary. I'm in my third year right now. So I'm going to give a talk about plant residues on stone tools from Oldupai Gorge in Tanzania. Basically, my discussion is based on how organic residues, especially of plants, can tell us about lithic function. My work is focusing on PhD, parts of my work, which I have just started with the samples I'm working on. And the sites I'm doing research are from eastern part of the gorge, which are Engona Ekiloki, Ndumoto Ekiloki, and Esoit and Ndumoto. These sites in the western part of the gorge are very useful, I would say so, because the part is well defined in terms of stratigraphy. And working with bed one archaeological materials, basically these are old one samples dating about 1.8 million years ago. Right now, the total number of samples which I'm having, or which I have started with, are 89 samples. So I have different objectives in my research. One is to identify stone tool function through micro residues, plant residues, as I have said before. And then I want to use plant residues to reconstruct paleo environmental conditions of this part where I'm working. Then I want to quantify the distribution of residues through geographic information system, the GIS. I also have research questions which I want to attempt in this work which I'm doing. One is to understand the changes, if there was any changes in tool use during 1.8 million years ago. And then I want to know, how does the proportion of residues change, spatial and temporal? Finally, I want to know if there is any correlations between tool type and tool function. As I've said before, that this is a first data set of my doctoral work. I have started working by using by sampling the materials I have by using random stratified sampling, whereby I have a pilot study which is basing on raw materials, and the percentage out of those 89 raw materials, I decided to use 15%, so that I can include as much as raw materials from these samples I'm working with. Now, by using stratified random sampling, you can realize that from one of the sites, which is Esoit and Ndumoto, I am showing you here 17 phonolites. Of course, I have different raw materials, but I choose to show you this slide by choosing just one raw material, which is phonolites. I have 17 phonolites in this site. Now, after applying this program, which is going to give me random samples, I calculate by finding what is a 15% of 17 samples. Basically, you find 2.55, which can be approximated to 3. That means I have 3 samples to choose from my general phonolite samples at Esoit and Ndumoto. And then you can see now here it is showing me 7, 17, and 13. Of course, these are randomized samples which the program has chosen for me. Now, I have, as I said earlier, I have 17 samples in total from Esoit and Ndumoto. Here, written phonolite, I have written these abstract numbers from number 1 to 17. But if you go back here, see 7, 17, and 13. 7 in red, 13 and 17. These are numbers which have been chosen. But in real sense, when I go to my general data set with the general raw materials, phonolite doesn't start as the first sample. You see number 7 here is 20 in terms of the sample ID, and number 13 is 33, and number 17 is a very last sample in my total samples of Aloyce and Dumoto, which is 51. What do I mean by here? If you go back here, I mean, if you come to this next slide, you see these are general samples from Aloyce and Dumoto. Number 1 is not phonolite, it is basalt. So if I'm to find which is the number 1 in my general samples when it comes to phonolite, you can see 007 is the phonolite, which starts as number 1. Now, as long as this program has told me to choose sample number 7, phonolite number 7, phonolite number 13, and phonolite number 17. Now, this number 20 in terms of ID is the one which is chosen as being number 7, and 33 is number 13. Next slide, I wanted to show you how now that, those samples which are chosen looks like. This is phonolite, which was a very last sample. In my slide, I showed you number 17 being chosen by this randomizer, which is in real sense, it is the very last sample, which is 51. And also I decided to show you this basalt from Aloyce and Dumoto, showing you that I have variety of materials. Variety of materials, of course. Next, I think these data sets which I have or samples are very unique. In what sense? Because the collection procedures they followed the uncontamination procedures or decontamination procedures. So what we did is to collect these samples in clean room. So we have under SDS, we do have mobile lab, as you can see here, now excavating in this and then using different, using the gloves, trowel, and clean room bags to make sure that we avoid the means of contamination when excavating these samples. Okay. Next, I wanted to show you how these 15% of raw materials apply. I do have Endumoto Ekiloki and Aloyce Endumoto. I wanted to show the frequency, sample frequency of each site. Now, the reason is to show you how 15% is much more inclusive when it comes to raw materials. In Oldupai, Gorge, these are, I would say, our main raw materials which are usually found at Oldupai. You can see basalt is there, chert is there, lava is there, phonolite, and quartzite. So these are the frequency materials which we expect to find them, and 15% was able to give me a variety of raw materials, as you can see. Next, in my next slide, I want to show you that basically with this research, what I do is I expect to have a sequential steps in my research. What I have described or I have mentioned earlier was one of the steps which I have done basically, and I expect to keep on doing other steps. That was techno-typological analysis, which I have done. I have all of the raw materials being typologized, and during that process of typologizing, I have been recording a time, meaning what time a certain material has been exposed, so that in any sense I can be able to talk about this scenario, which is much more important in scientific analysis. Also, after this analysis, I expect to start plotting of residues. As you see here, it's me in our clean room using microscope plotting residues by using microscope. After residues are plotted, my next step is expected to be extracting, consolidating, identifying, and classifying those residues in terms of morphotypes, etc. Next step is expected to be now the use of geographic information system to map, doing plot maps. The plot maps will help me to show, to have a good visualization of which part of the sample, which part of the lithic was used to process certain raw materials. Now, with this image, I wanted to show you how the plotted part is being seen. What you see here, these are the shapes of plant residues basing on our shape legend. So what you see is just one shape. I could have different shapes if I could have different residues in this part of the stone. So you see one shape because what I can see when plotting these residues is just one type of residue. At this right side, all you see is how now the residue is seen under microscope when you use 50 magnification. I wanted to share with you how plot maps looks like. After applying GIS, now I have this part which was specifically used by any activities, but what you find is residues, distribution. Now the plot maps helps us to understand the spatial distribution, as I've said before. So what is the significance of this study? One, is to reconstruct past human activities or behaviors, which is much more to understand the use of stone tools in Oldupai Gorge, especially in this part of the gorge which I'm doing my research. Next thing is to understand what might have been the ancient vegetation cover in this part of the research. And then I expect to reconstruct the environment of ancient past. And one of the other very crucial thing is for proper curation of lithics, which are housed in the National Museum of Tanzania. The reason to why I could think about proper curation is because always researchers will want samples or lithics which are well curated, which are well stored, which are following the procedures of decontamination for further research. And the reason to why I could write or talk about NMT, which is a National Museum of Tanzania, it's because I'm affiliated to this institution where I'm working as an archaeology curator with this institution in Tanzania. So basically, this is that was my last slide, which was a bit short, but so thankful for everyone who could listen to my presentation. Thank you.
All right, we have a couple questions now, so I'll just display those in the chat. I'll read them out first and then I'll post them here in the Zoom chat. Okay, so the first one is, how long does identifying these residues usually take?
Okay, first of all, should I stop sharing?
You can leave it on the screen, it's fine.
Okay. This question is, of course, it's very tricky. What I would say, I don't really have a time frame. To say how long, but one thing is real. It takes so long even to finish or to complete one surface of the stone. I worked with Mr. Fragas, who is asking this question. He knows how long we took to finish even one surface of the stone. It was, I would say almost 3 days. You can do that.
Oh yeah,
doing their first. Doing just one surface of the stone.
Yeah, totally. Yeah. Okay. Next question. How can plant residues of anthropogenic origin be differentiated from plant residues of natural origin?
Yeah, it's a great question, which is, I mean, it has always been imposed to understand how one can differentiate this. One of the good methods is when we use GIS, it helps a lot to understand which part was mostly used, you know, but this is not enough. What you can do, you always have to follow procedures like having, we have the so-called control samples. Once you have a control samples, let's say you are doing research somewhere, you have a stone tool, which you excavate, you always have to look at the matrix of that stone. You find the segments, you do research by extracting residues from those segments. Now, once you have that, you can do a kind of correlation whether those, to understand whether those residues came from stone and then to sediments, or they just belong to that stone tool, which will tell you that it is very true that these are not, I mean, these are from use, not, yes, these are from use residues, not from natural residues.
Okay, another question just came in. I'll display it right now. Do you believe that meat played an important role during the older one?
What?
Do you believe that meat played an important role during the older one?
Of course, I would say so, because by following different researchers, they have talked much about evolution. You know, when they talk about human evolution, we can speak about technological advancement as one of evolution stories. So in terms of technological advancement, you expect to find the much more modified stone tools. So if you find those much more modified stone tools real, you're going to say there was a kind of transition in terms of brain increase and so forth. Now, reading about brain expansion and the brain, I mean increase or wherever, I have researched and found that meat played a great role for brain advancement. So I would say yes.
Okay, at the moment we don't have any more questions, so I'd just like to thank you, Aloyce, for giving a fantastic talk. Very interesting stuff, and I'd like to wish you much luck in the completion of your PhD, and I'd like to thank our general attendees for tuning in, and we'll see you in the following sessions.
Thank you so much, Mr. Felix.
No problem.
See you.
See you.